Dr. Krista Rogers obtained a BS from UCLA in organismal biology, ecology, and evolution. She then received her PhD in developmental biology from Georgetown University under the mentorship of Dr. Elena Silva Casey. In 2010, she moved to a postdoctoral position at the California Institute of Technology in the Division of Biology and Biological Engineering in the lab of Dr. Marianne Bronner. In 2016, she became an assistant professor at the California State University Northridge in the biology department, where she created a thriving undergraduate research program in developmental biology. In 2019, Dr. Rogers moved to UC Davis School of Veterinary Medicine and is now an associate professor in the Department of Anatomy, Physiology, and Cell Biology. Her lab studies the molecular mechanisms that drive neural crest cell development in chicken, quail, peafowl, and axolotl embryos. The lab focuses on identifying and characterizing conserved and divergent genes and proteins involved in the specification, migration, and differentiation of neural crest cells across species. We are so glad to have you with us today. Please take it away.
Thank you so much for that wonderful introduction. I feel like I don't have to really talk about anything because you sort of, you told the whole story. So thanks so much for inviting me to speak here. I've been following the seminar series, and it's been really fun to see what everybody's doing and all the different types of imaging and bioimaging that people are doing. So today I'm going to tell you two short stories that are happening in my lab, each with hanging ends that we're still working on. But before I get into the science or into the depth of the science, I just want to give you an overview of what my lab does. I see. You spotlighted me. Okay, so my lab is a developmental biology lab. And so what developmental biologists do is we try to understand the processes and the mechanisms that allow a single cell to undergo molecular and morphological changes to become a more complex embryo, and then how that changes to become an adult organism. And development is this ongoing process where we study changes in organisms across time. But to understand all of these processes it's a really big question to ask. I can't focus on understanding how every organism is formed from a single cell because it's a very big question. And so what my lab studies is the formation of ectodermal cells. And so just to take you back to some of the basic developmental biology, what you're looking at here is a chicken embryo with its head into the screen and its tail out, and it doesn't look like a chicken because it's a gastrulate stage embryo. So gastrulation is the most important time in development. It is where the three germ layers form. Those germ layers are endoderm, mesoderm, and ectoderm. And the one we study is ectoderm. In my personal opinion, it's the most important germ layer, but ectodermal cells form the central nervous system, so the brain and the spinal cord. They form the skin and sensory organs, and they also form neural crest cells. So what you're looking at here is a depiction of this flat layer of ectoderm, where this region marked in blue will form the neural plate, which will become the neural tube and the central nervous system. And then neural crest cells form in this green region here called the neural plate border. So focusing on neural crest cells is really important because these cells are very dynamic. They undergo molecular changes that allow them to start out in the developing central nervous system. They undergo a process called an epithelial to mesenchymal transition, where they go from being really tightly adherent to collectively migrating to very mesenchymal and invasive. The mechanisms that drive these different processes in the embryo are reused in disease states like cancer for cancer metastasis. So if we can understand what's happening in developing embryos, not only will that give us insight into how these different cells and tissues form, but it will also help us understand how things might be going wrong in disease states. So what you're looking at here is a developing chicken embryo with the head to the top, and these green cells are neural crest cells. And they start out in the midline, and you can see they spread around the head. And in this case, these are cranial neural crest cells, so they're going to form the face. They will come around and migrate and form things like the craniofacial bone and cartilage. But if we take a step back and try to figure out what are we looking at here? If we take a bisection or a transverse section of this developing embryo and we rotate it 90 degrees, at the earliest stages of development, you can see that there's this flat tissue layer of ectoderm. The middle becomes the neural plate, which will become the central nervous system. And these neural plate border cells form on either side of the developing neural plate. As the neural tube forms and rolls up and fuses, neural crest cells are specified, or they become neural crest cells. And they upregulate specific genes that mark these cells in the dorsal neural tube. So this is dorsal. And in this case, these cells are then going to undergo multiple stages of delamination, collective migration, and mesenchymalization, where they become very invasive as development proceeds. So at this developmental stage, you can see these cells are starting to change, and they're a little bit different from the blue marked cells. They change their cell-cell adhesion, they change the different transcription factors that are expressed. And then what that does is it allows them to leave that tissue and then migrate throughout the embryo to distant sites. Neural crest cells make more than 30 different adult tissue and cell type derivatives. And so some of the examples are they make the pigment cells, they make the sensory nervous system. So they make the peripheral nervous system and the enteric nervous system. They make craniofacial bone and cartilage, smooth muscle cells, fat cells, you name it. You can imagine that all of these different cell types that neural crest cells can make in a developing embryo are very important. And what we know is that if there are any perturbations or developmental complications that occur at the earliest stages or even at the later stages, we can then lose some of these different derivatives, or it can affect their development. So in the case that neural crest cell development goes awry, neurocrestopathies can form. So neurocrestopathies are pathological disorders that are linked to abnormal neural crest cell development, and those include things like cleft lip or palate, or facial palsy, or albinism, deafness. There are a number of different neurocrestopathies. But what's really interesting to know is that since neural crest cells are a vertebrate-specific cell type, that means that neurocrestopathies only occur in vertebrates, but it also means that neurocrestopathies can occur in any vertebrate system. And so the work we do is relevant to animal, human development, et cetera. And so just to give you an overview of what I'm going to talk about today, I always go back to the central dogma of biology, which is DNA is transcribed into RNA, which is then translated into protein, because it gives you a sense of the sort of levels that we study in the lab. So if you have your DNA, that's your roadmap for what's going to be expressed in a certain cell. It's transcribed into RNA. And the first part of my talk is going to focus on transcription factors that control the formation of neural crest cells, their migration, and changes in cell-cell adhesion that allow them to become the different derivatives that they're going to become. After that mRNA is translated into the proteins are folded, and the proteins are the things that do the work. And so a lot of my studies focus on changes in protein localization, expression, et cetera. So here's the protein doing the work. But what we know is that proteins are also worked on. So after a protein is folded and it's doing whatever it's supposed to be doing, there are post-translational mechanisms that can traffic these proteins around the cells, move them to where they need to be. And so I'll be telling you a little bit of one of our transcriptional stories today and also one of our post-translational stories. And I will close my window because there's trucks. Okay, so the major questions in my lab are to understand the genetic and molecular mechanisms that control neural crest cell development. And I use these two examples. This is my dog Roxy, and this is a collie with a long snout. And the reason I use these two examples is because these animals are the same species, and neural crest cells make their craniofacial regions, and yet you can see there's a lot of variety in the structures that can be created using similar roadmaps or similar recipes. And so we're interested in understanding even within organism variation, but we also want to know how do different organisms form different craniofacial structures. So we use multiple organisms to do comparative analysis to understand whether or not the mechanisms that control neural crest cell development are conserved or if they're species specific. And so you can see here, this is a chicken, this is one of our models, and here's an axolotl, it's also one of our models. And their craniofacial structures are not only different shapes, but different consistencies, hardness, et cetera. And so we can focus on the molecular mechanisms that control the formation of these cells and understand how they, where the paths diverge to make these different structures. And then our lab also studies how environmental exposures can affect embryonic development and the formation of these neural crest cells, and theoretically how environmental exposures can affect the development of neural crest structures like the craniofacial bone and cartilage. To answer these questions, we use 4 different models, as Vanessa mentioned. We use Gallus gallus, or the chicken. We use Coturnix japonica, or the Japanese quail, Pavo cristatus, or the Indian peafowl, and Ambystoma mexicanum, or the axolotl. And the reason we use so many different organisms is because it allows us to do direct comparisons within species for very similar animals like birds, or also across species and across different families and groups and orders, et cetera, so that we can see how different these developmental processes can be. Now, of note, most of our functional studies occur in chicken and quail because of these 4 organisms, their genomes are both annotated and sequenced, and so that allows us to do functional perturbation. And then we also use multiple modes of imaging in our lab, but focus a lot on fluorescence microscopy, everything from basic fluorescence microscopy to light sheet imaging and confocal. So, to get into the science, the first thing we wanted to understand is how similar or different developmental processes are across species. And so to do this, what we did was a study that I'm not going to talk about because it's already published, but I'll give you the highlights and the take-home message. And we had a group of people who worked really hard during the pandemic to try to understand, is the developmental timing the same across different bird species to form neural crest cells? And so we wanted to focus on looking at whether transcription factor expression, onset, and localization was conserved. So what we did was we focused on this very complicated map called the gene regulatory network. So the neural crest gene regulatory network is essentially a recipe. And if our recipe is meant to make a cake, then we have all of the ingredients. Those ingredients have to go in at a certain time, and then things have to happen at the right time and in the right place to make that cake. Well, in this case, the neural crest gene regulatory network makes neural crest cells, and those neural crest cells make their different derivatives like cartilage, bone, or pigment cells. And so what we know is that in certain organisms, there's the onset of gene expression and protein expression, and those proteins can then activate or downregulate their targets, and then that leads to this cascade of different changes. And that's essentially what the gene regulatory network is a recipe that has been identified based on studies across multiple organisms that focus on functional perturbations to understand when and where these factors are necessary for the formation of these cells. But what I'm going to focus on are the migratory and premigratory cells. And so thinking about what we know about the neural crest gene regulatory network, we don't know whether or not the spatiotemporal expression of each and every one of these factors is conserved across species. We know that it's generally conserved across our 5 main model organisms, but we don't know whether or not emerging and unique model organisms still maintain this, or even if the time is the same, because it's really hard to compare stage to stage from very different organisms like amniotes, mouse, chicken, for example, versus anamniotes like zebrafish and frog. We also don't know whether or not a lot of these direct interactions where one protein activates the expression of a gene is actually conserved across species. And so it's really important to do comparative horizontal integration of these studies to know whether or not this is conserved. And then we also want to know whether these proteins that are expressed across all of these different cell phases, have the same function. So for example, if you see these proteins marked in orange, these are the same proteins that are marked in pink here. And in pre-migratory neural crest cells, those cells are still really tightly adhesive. They have not left the neural tube yet, and they co-express a lot of different factors. But then they leave the neural tube and they become invasive and they start migrating, and yet they're still expressing these same transcription factors. And so we're interested in finding out whether there are stage-specific functions of each of these factors. But the first thing that we wanted to do was really just compare and contrast gene and protein expression. And so what we did was we used in situ hybridization chain reaction and immunohistochemistry to compare the developmental timeline of neural crest cells in quail, chicken, and peafowl embryos. And what we found was that quail embryos have this massively, a very rapid onset of neural crest cell development. And what you're looking at here is essentially the expression of a protein, for example, or the fact that it's turned on based on immunohistochemistry. And if you compare these graphs, these are the stages of development, and this is quail on top and chicken on the bottom. And what we found is that very unlike studies in other model organisms, the transcription factor SNAIL2 was expressed in the earliest developing neural crest cells, neural plate border cells even. And these cells are very epithelial, and the known role of SNAIL2 didn't seem to be doing what we thought it was doing in quail, but in chicken embryos, that is the model that most of these studies have been based on. So this was very stereotypical, that you have the neural crest progenitors here that then upregulate these different transcription factors. Sorry, I've had a lot of coffee this morning. Upregulate these different transcription factors one after the other. But in quail embryos, there was co-expression of the factor SNAIL2. So keep that in mind because that will come in play later in this section. But what we know is that these cells seem to be different, the environment's different, but yet they're not migratory. So in the quail embryos, neural crest cells form earlier, and we identified that they do migrate earlier, but the developmental time point is very similar to that in chicken. And so we were trying to find out what is happening in these cells, why are they migrating earlier, why are they more invasive? And we wanted to know whether or not differential expression of these factors actually matters for development. So to answer that question, we focused on the very last transcription factor that is activated in the gene regulatory network, the protein SOX10. So SOX10 is turned on just as these cells are migrating. It's one of the major markers of neural crest cells that everybody uses to mark the population of these migratory neural crest cells. And prior work identified that SOX10 is a migration driver, and it drives migration theoretically by activating the expression of these very migratory cadherins, these cell adhesion molecules that allow for collective and mesenchymal migration. So when this protein or this gene and protein was overexpressed in previous studies, they identified that if you take chicken embryos and you put this SOX10 in the spinal cord, what it does is all of these cells have too much SOX10. They migrate out of the neural tube to the point where the spinal cord is no longer a spinal cord, and it now is just a bunch of migratory cells. If you compare that to GFP, these cells are forming what they're supposed to be forming, the spinal cord, the dorsal root ganglia, the ventral root, but the cells that have too much SOX10 migrate. And when they did these similar experiments in frog, they also identified that you have this expansion of the neural crest cell population and cells that are migrating. And so we wanted to confirm that this was actually a study based on the idea that SOX10 is going to have the same role across species. That was our hypothesis. That's not what happened, but the idea was this is going to be a really easy comparative analysis so that when we move on to the other factors that are more upstream in the pathway, this one at least should be conserved. So what did we do to understand whether SOX10 is conserved across species? We took SOX10, full-length SOX10, and we overexpressed it at very early stages of development. Looking at the quail here, you can see that at the stage we overexpressed it, again, these cells are slightly different because they have both PAX7 and SNAIL2, so they are neural crest cells or neural plate border cells, but they have different potential. And then when we overexpressed it in chicken embryos, those cells only have one of these factors. And what we did was then incubated the embryos. We can inject and electroporate them at early stages, incubate them, and collect them at the stages that we're interested in. And the question that we had is SOX10 sufficient to drive migration? Are we going to see that SOX10 is actively driving migration in both quail and chicken embryos, or is it going to drive differentiation? Is SOX10 actually doing something else? And so these experiments are slightly different than the earlier ones because they're in a different axial region. They're in the head versus the trunk. And also overexpressions are at a slightly earlier stage, so the cells are more stem-like. So when we overexpress SOX10 in the chicken embryos, what we found is we had this ectopic expression of the factor SOX9. So SOX9 is a neural crest specifier. It's only supposed to be upregulated at these later stages here in the dorsal neural tube, but every single cell that has SOX10 also had SOX9. But in contrast, when we did the same experiment in quail, we found that in fact, SOX10 was sufficient to drive migration. And so what we're seeing here is looking at this simplified gene regulatory network, SOX10 is sufficient to drive cell migration or neural crest cell migration in quail embryos, but it doesn't appear to be sufficient to drive neural crest cell migration in chick embryos. And so we wanted to look at other neural crest markers to actually identify whether or not this phenotype was occurring across all neural crest cells. And what we found is that when we overexpress SOX10 in chick embryos, there was a marked cell autonomous reduction of PAX7. So PAX7 is a neural crest progenitor marker. The cells continue expressing that until they migrate, in which they downregulate it. And in chick embryos, you have this reduction. Everywhere there's SOX10, there's a loss of PAX7. So what we're seeing is SOX10 is activating SOX9 in chicken embryos. So you have this upregulation of this factor, but the other neural crest markers are reduced. But in contrast, in quail, the entire population of neural crest cells was not only expanded, but these cells were migratory. And so what we're seeing here is that the same treatment, overexpression of SOX10, seems to be driving different responses in the chicken versus the quail. Now, these are two really closely related organisms. And so what it made us think about is the idea that maybe even in closely related organisms, there are slightly different recipes to get the same outcome. And so we know that in terms of the quail neural crest gene regulatory network, the expression of SNAIL2 here is much closer or even overlapping with that of PAC7. And so the timeline is shifted to an earlier stage here, and these cells are likely primed to migrate. So when we overexpress SOX10, we're actually activating this pathway and driving migration. But in contrast, in chick embryos, the SNAIL2 expression is much, much later. And so when we overexpress SOX10, it's actually happening before we ever see SNAIL2 in these embryos, and it seems to be driving potentially differentiation of these cells into some other cell type. So to understand what these cells are differentiating into, what Raneesh did was he actually analyzed a single cell data set from mouse embryos. And so there are a ton of beautiful single cell data sets out there that track the trajectory of neural crest cells and define what they become and which markers are expressed in which time. And in this paper from the Adamaco lab, what they identified was that SOX10 positive neural crest cells tend to make a host of different derivatives shown here. But when Raneesh mined the data set to try to understand what the cells that we were seeing in chick embryos were becoming, he focused on the factors that we saw. When we overexpress SOX10, there's ectopic SOX9 and SOX2 and CDH2, which is cadherin 2 or N-cadherin. And the only cells in the Adamaco data set that seem to have those markers expressed were proliferating Schwann cell precursors. And so what we did was we then did immunohistochemistry for Schwann cells. So Schwann cells are support cells that sort of become like myelin-based support cells for the peripheral nervous system. And although you can see on the control side here of this is the uninjected side of a chicken embryo, P0, which is a Schwann cell marker, is expressed in migratory neural crest cells. In the embryos that had ectopic SOX10, it seems to be expressed within the developing neural tube and also upregulated in the migratory crest cells. So in chicken embryos, SOX10 overexpression is not driving migration. In fact, these cells don't seem to be able to migrate, and they're differentiating where they stand into Schwann cells. So that's really interesting, but we really wanted to know why this is happening. And so we went back to our main love in the lab, and I would say that 90% of our stories end up at cell adhesion. And so when we think about how neural crest cells go from being this very epithelial cell type within the neural tube, and how they then detach from the neural tube and migrate collectively away from the neural tube, how they become invasive, the first place I think, the first place I go is changes in cell-cell adhesion. And so what you can see is that when we think about cell adhesion, our lab focuses on cadherin proteins. Cadherin proteins are calcium-dependent cell-cell adhesion molecules that are transmembrane proteins that interact across cell barriers or in the extracellular space with adjacent cadherins to maintain these epithelia. And when we did immunohistochemistry for these different markers, you can see they have overlapping and discrete expression. So CDH2, which is N-cadherin, seems to be expressed in the neural tube, but is absent from neural crest cells. But in contrast, these other cadherins, both type 1, which are the epithelial types, and type 2, which are the migratory types, are all upregulated in neural crest cells, either during the process of EMT or during migration here. And what we know is that cadherins are well characterized factors that regulate morphogenesis, cell migration, and developmental processes. And mutations in these proteins alone can cause developmental anomalies. And so our work in cadherin proteins has really focused on what happens if we add some or take them away. So going back to the recipe analogy, if you add extra flour, what happens to the final cake? And so what we've done in the past is really do these gain and loss of function assays where we knock down cadherins or we add cadherins. And what we found is that the cadherin proteins are very tightly regulated. And if you perturb one, the others respond. And so our previous work identified that if we knock down one cadherin, we get more of the other. And if we overexpress that same cadherin, we lose the other, which means that these are reciprocally fighting for available space on the membrane and available factors. And we know that when we do these different perturbation experiments, you add something, you take something away, we always lose neural crest cells. And the cells can't be specified, and when they are specified, they don't migrate appropriately. So with that background, we went back to our SOX10 studies to try to figure out in the chicken embryo, why are these cells not migrating when we overexpress SOX10? And we wanted to focus on understanding cadherins. And the reason for this is that in normal neural crest cells, if you look at the central nervous system here, this is neural crest, and they collectively migrate as a group away from the developing neural tube. You can see if the video is playing, you can see that they're sort of migrating consistently away. But when we have too much of type 1 CDH1 or E-cadherin, these cells just sort of stay and they don't migrate. So we hypothesized that SOX10 overexpression in the chick embryo is driving an anti-migration cadherin profile, but in the quail embryo, it seems to be driving the pro-migration cadherin profile. So we did immunohistochemistry for CDH1 or E-cadherin in chick and quail to validate that these proteins are actually localized to the same place during development. And we found that yes, ECAD is expressed in the neural tube and the collectively migrating neural crest cells in both organisms, or CDH1. And when we overexpress SOX10, we have this cell autonomous loss of CDH1 in the chicken embryo. And in quail embryos, we have this gain, or at least a maintenance of CDH1. So we know, based on studies in frog and chicken embryos, that CDH1 is necessary for collective migration, and it actually can promote collective migration out of the neural tube, as you see here. And so when we lose it, those cells are not permitted or not able to collectively migrate out of the neural tube. Whereas these quail cells seem to have the expression of E-cadherin that they should have, and so therefore they're capable of migrating. And so here we can see drastic differences in the response of SOX10 gain of function, that when we do it in quail, CDH1 seems to be upregulated and those cells are collectively migrating. But when we do it in the chicken, the protein is lost and those cells are unable to migrate. But that's not all. We also have this type 1 very epithelial or adherent cadherin, CDH2, and this is actively removed from neural crest cells because it's got such a strong adhesion force that if it's expressed in neural crest cells, they're unable to leave the neural tube. And so we can see that it is in the neural tube and removed from the neural crest cell and cells in both organisms. And when we overexpress SOX10 in chick embryos, we found that there was cell autonomous increases, meaning every cell that had SOX10 had excess N-cadherin or CDH2. So not only are these cells unable to collectively migrate out of the neural tube, but they also now have upregulated expression of this really tightly adherent cell adhesion molecule, which means that they're actually unable to migrate at all. And so when we are getting the phenotype in chicken embryos that SOX10 seems to drive differentiation and these cells are forming Schwann cells, it's likely because they're unable to migrate, and so they do the next best thing, which is differentiate. But in contrast, in quail embryos, these cells have lost this really adhesive cadherin. So not only do they have the expression of the collectively migrating force, but they've lost the thing that's preventing them from migrating. And so what we think is happening is that at a transcription level, so SOX10 is a transcription factor, we are seeing that SOX10 alone can drive or is sufficient to drive migration in quail, likely because of these effects on the cadherins. But in chick embryos, we have really anti-migration profiles in the presence of SOX10. And so what Johnny in the lab did was he wanted to see if he could rescue that phenotype. So what we know is that SNAIL2 is upregulated at an earlier stage in quail embryos, and that this environment is different. And so what he did was he said, well, what if I take the SNAIL2 in chicken embryos and I overexpress that at the same stage that I overexpress SOX10? So now I'm adding both of these factors to chicken embryos to see if I can rescue the migration phenotype. And what he found is that, in fact, when he overexpressed both SOX10 and SNAIL2 together in chick embryos and recreated the quail-like environment, he was capable of not only rescuing the loss of PAC7, but now these cells were migrating. And so what we think is happening is that the environment matters and timing matters. And I think that this is a really good example of how very similar organisms might use different recipes to get the same outcome. And in this case, we can quailize the chicken embryo by adding SNAIL2 and by changing up the environment. So what we're doing on this next is actually trying to identify what is it that SOX10 is binding to. And so what Julia is going to be working on is really defining the different targets in quail and chicken embryos to define, is it transcriptionally regulating the expression of one of these cadherins or multiple? And is it working alone, or is it interacting with something like SNAIL2 to do these things? And so knowing that N-cadherin is expressed at the gene or the transcript level in a very similar profile as it is the protein level, she's going to be focusing on trying to understand if SOX10 is maybe inhibiting N-cadherin or CDH2 directly. And so this is the first part of the talk. The second part's a little bit shorter, but ultimately what I wanted you to take away from this first part is that at the transcriptional level, this neural crest gene regulatory network that we think about might differ in timing or in partners during development, even in similar species. And so it's really important that we expand our model organisms rather than condensing them and try to understand how these animals organize and alter these molecular processes to get the same outcome, which is ultimately a face. So knowing that proteins do the work and that these proteins are shuttled around, the second thing I want to tell you about is a project that we're looking at to study cadherin proteins and how these proteins are moved around the cells. And so the work that I'm going to show you has been mostly done by Camilo and Bernice. And what we're really trying to understand is how do you get these dynamic changes in protein localization across multiple stages? So what you have here is a cross-section of an embryo. This is the neural crest cell region, and you can see the neural crest cells are marked by these dashed lines. And we know that N-cadherin at the protein level is completely absent from those cells. We know that E-cadherin or CDH1 is upregulated. And we know that cadherin 11 is upregulated in these EMT stage, these cells that are delaminating and collectively migrating. So we have these different expression profiles. But what's really interesting about these cadherins is that if you use HCR and do in situ hybridization and look at the localization of the transcript, the CDH2 that I just mentioned looks very much at the RNA and the protein level. It looks the same. So you can see something's probably transcriptionally downregulating CDH2 in this region to allow for neural crest cell delamination and migration, and that's reflected in the protein. But in contrast, E-cadherin is not expressed really at the transcript level. And we've done these analyses using qPCR in single cell data sets and also using HCR, and we just don't see the transcript, and yet we see this protein, suggesting that it might be post-translationally regulated, that maybe the protein is stable and it's being pulled and moved towards the membrane as needed. And so what we hypothesized is that if you have these cells, you have these epithelial cells, and they have expression or localization of specific cadherins to allow them to stick to each other. During EMT, when the cells are really changing their polarity and cell-cell adhesion, this microtubule network is trafficking these epithelial cadherins away from the membrane so that they can become more flexible and mesenchymal. So that was our hypothesis. So we first focused on the role of TUB3. So beta-3 tubulin is thought to be a neuronal marker. You can see here in the chicken embryo, it's expressed in these nerves, the trigeminal ganglia, and there are a number of different nerves you can see that express it. But we had previously identified that TUB3 is also upregulated specifically in neural crest cells. And if you look at the single cell heat map, basically there are multiple beta and alpha tubulins that are expressed uniquely in neural crest cells and that are upregulated during the specification and EMT stages. And so when we look again at the transcripts, you can see that TUB3 itself is upregulated in these neural crest cells here in the dorsal neural tube, and that's reflected at the gene and the protein level, or the transcript and the protein level. And so we thought, okay, so if microtubules are necessary for trafficking cadherins to and fro, then TUB3 is going to be our protein of interest. And you can see that it's upregulated at the same stages that these markers for neural crest cells are also upregulated. So we're thinking that there's a link between neural crest cell EMT and TUB3. So what is TUB3? TUB3 is a beta subunit of microtubules, and microtubules are these dynamic cell skeletal proteins or cytoskeletal proteins. We know that they can interact with both actin filaments and also cadherins, and that in fact, cadherins can be trafficked to and from the membrane on microtubules. We also know that if there's mutations in TUB3 specifically, it can cause developmental anomalies, including things like congenital fibrosis of the extraocular muscle, which causes blepharoptosis, cortical dysplasia or absence of brain growth, and a number of different syndromes. So we know it's important. And we know that some of these disorders can be linked to tissues that are derived from neural crest cells. So what we did was we wanted to see, well, is TUB3 necessary for neural crest cell migration? If we think that this protein and its microtubules are the ones that are trafficking cadherins, and we know that cadherins are necessary for these processes, let's take it away and see what happens. Again, we're altering the recipe. What we did was we knocked down TUB3 using translation blocking morpholinos, which means that the RNA is expressed and then we prevent its translation into protein. And we did this solely in chicken embryos. Well, we did this for this part in chicken embryos. And you can see that the onset of TUB3 here in yellow is actually after the stage at which we knocked it down, so it doesn't ever come on. And when we knocked it down, we found that there was an expansion of the neural crest cell population, and they migrated more rapidly. And not only that, but these cells seem to maintain this epithelial cadherin, so they have e-cadherin. Now, based on the study, the first part of this story, where I mentioned that e-cadherin or CDH1 is not only necessary for collective migration, but it seems to drive collective migration, these data track that in the sense that we've removed TUB3, now you have this maintenance of e-cadherin on the membrane, and that's allowing these cells to migrate very fast. So we know that TUB3 seems to be necessary actually to remove e-cadherin from the membrane, which is a little bit different than what our hypothesis was. So we did neural tube explant assays. So what we can do is we can cut out the developing neural tube, we can culture it ex vivo, and you can see neural crest cells intrinsically migrate out of these on a flat dish. And what this allows us to do is really get a sense of cell structure, cell morphology in our wild type and perturbed cells. And you can see that this is the neural tube. It's very epithelial, little round nuclei that express SOX9. And as neural crest cells migrate, they go through a collective migration stage where they're sort of together, and then they mesenchymalize and they become very invasive. And that's reflected at the actin level. So actin is in epithelial cells, very peripheral and membrane bound, but in mesenchymal cells, it's very polarized. And you can see that it's expressed both in the leading edge and the trailing edge of the cell. When we knock down TUB3, we have different nuclear structure, and also the actin is very peripheral and it's membrane bound. So what this means is that in the absence of TUB3, neural crest cells maintain their epithelial or collective nature. And because they have the expression of that cadherin molecule, they also are linking that cell membrane to the actin cytoskeleton. So basically these cells are very epithelial, but also very invasive. And we didn't understand why they're able to migrate so efficiently until we looked at the type 2 or migratory cadherins and found that in the absence of TUB3, there's an upregulation of both cadherin 7 and 11, which drive migration. And so what we have is this completely wonky neural crest experience where these cells aren't trafficking the cadherins to the right places at the right time, but rather than losing neural crest cells or inhibiting migration, we're adding to both. And so we altered our hypothesis and hypothesized now that TUB3 mediated trafficking is necessary to move the migratory cadherins to the membrane and remove e-cadherin from the membrane to allow the cells not just to collectively migrate, but to mesenchymalize and separate from each other. And so to determine if TUB3 is sufficient for this process, we added it. So rather than taking it away, we did overexpression. And what we found was when we looked at e-cadherin, the cells that had excess TUB3 have this massive increase of e-cadherin, and also it's driven to the cytoplasm. So if you look at cells on this left uninjected side that are dividing, e-cadherin is maintained on the membrane. But in contrast, in the cells that have excess TUB3, there's a ton of e-cadherin in the cytoplasm, suggesting that its presence alone is sufficient to traffic e-cadherin from the membrane to the cytoplasm, because we've already established that it's not really transcribed, so this has to be a post-translational mechanism. So we also identified that in these same cells, they're losing neural crest cells. So in contrast to the knockdown where you gain neural crest cells, overexpression of TUB3 reduces the neural crest cell population. And surprisingly, or well, I guess unsurprisingly, there's not really an effect on N-cadherin. So TUB3 mediated trafficking is focused really on E-cadherin rather than N. And so what this means is that while N-cadherin is likely regulated by transcriptional changes, E-cadherin is likely regulated post-translationally by moving it around the cell. So we also identified in these embryos that had a loss of, too much TUB3 and they have the cytoplasmic E-cadherin, there's a really strong increase of proliferation. So every cell that seems to have TUB3 also has the expression of phosphorylated histone H3, which marks cells that are in mitosis. And the increase was pretty impressive, honestly. And then we let these embryos grow thinking, gosh, are they going to have, really big neural tubes? And what ends up happening is that the neural tubes divide through the apical side of the neural tube, and they create solid neural tracts. So they're no longer tubes, but they're rather neurogenic tracts of cells that ultimately end up dying. So rather than having a spinal cord that has a ventricle, it's just a spinal rod. And the neural tube in the cranial region divides so much that the cells become very tumor-like, and they sort of split the neural tube in half. And so all of our cells all of their embryos end up with these holes in the head. So overexpression of TUB3 does traffic cadherin at the molecular level, but the outcome for the embryo is very bad. And when we started looking into this, it's apparently very well known and established that cytoplasmic cadherin and excess TUB3 seems to drive metastasis, cell proliferation, and in fact, worsening symptoms in cancer. So what we're doing next with this study is that Camilo is proceeding to try to understand whether or not the role for TUB3 and post-translational protein trafficking is conserved across species. And what I can show you in our preliminary data is that, in fact, it is conserved. So when we do these same experiments in quail, when we're perturbing TUB3 and microtubules, we seem to have that overexpression of TUB3 in quail does drive cytoplasmic cadherin, or CDH1. It does increase proliferation. And in fact, what this tells us is that although at the transcriptional level, there might be divergent mechanisms to drive the formation and migration of these cells, when you get down to it at the protein level, the mechanisms seem to be the same. And we also have this reduction of neural crest cells in quail. So this is very conserved. And so what Camilo wanted to do is that he worked with Nancy, who was our summer student, and they wanted to basically originally the plan was to define TUB3's partner subunit, but that is not what the project ended up being, as it is with science. What we ended up doing is characterizing all of these different tubulin subunits across multiple stages of development, and we've identified that, in fact, this is HCR in multiple stages of chicken embryos, but in fact, what we've identified is that at different developmental stages, each of these developing tissues, whether we're talking about ectoderm, mesoderm, endoderm, there are tissue-specific expression of tubulin subunits. So in this case, for example, TUB6 is expressed in this myotome that's going to form the skeletal muscle. But Nancy has done some really beautiful HCR. And so now we know that although we haven't identified the partner of TUB3, a lot of the beta subunits seem to have very specific tissue expression during development. So that's going to be a really fun adventure that we have, trying to figure out what they're all doing. And then, so we also have Joanne in the lab, and it turns out microtubules are the highway, but you cannot traffic cargo without vehicles. And so the vehicles are motor proteins. And so what Joanne has been doing is trying to characterize and identify which of these motor proteins is necessary for trafficking the cadherins. And so she's identified some really pretty possible targets that we're looking at that are expressed at the right time and place to be trafficking these molecules, but that's TBD. There's a lot of stuff there, but ultimately all roads lead to beaks. So what does that mean? It means that when we look at the different transcription factors, there might be different timing of onset. Neural crest cells develop at different rates. There's heterochrony in the expression of these different genes and proteins across bird species. But ultimately, the goal is to get a beak, and to get this very specific beak structure, you have to have limitations. And so in this case, at the post-translational level, the localization of cadherins is tightly regulated by microtubules and trafficking. And that TUB3 role is very specific to get at the cadherin where it's supposed to be at the right time and at the right place. And what this tells us is that there might be flexibility at some parts of the program, but at others, there has to be physical and molecular constraints that have to control how many cells you have, how they migrate, et cetera. And so just to summarize what I've told you today, we know that SOX10, the transcription factor, drives differential responses in chicken and quail, that we see the differential responses in what the cells become and how they behave based on differential cadherin expression. We know that when we knock down TUB3, we end up having this increase of the number of collectively migrating neural crest cells. And in contrast, when we overexpress it, we tend to drive proliferation and we lose neural crest cells. But ultimately, both gain and loss of cadherins, which is where both of these projects ended up, causes abnormal neural crest cell development. And so a lot of what we're working on in the lab right now is trying to understand the outcome for the embryo. How is beak structure affected, which would then be analogous to how craniofacial structures are affected. When we do these early perturbations, what happens at the end? And so with that, I would really love to thank my lab. They've done so much beautiful work, and our funding sources. And again, thank everybody for coming, and especially during the summertime, heat of August. And thank you for having me, and I'm happy to take any questions.
That was fantastic. Thank you. I have a question as we're letting everybody get their questions together. So in the movies, when you were showing with Soxtan and the cells were, you were saying they're not migrating out because of the cadherins that are stuck together. Do you see any like changes in their directionality, like you see in developmental movies where they move, but they're not really moving together as they should?
Yeah, so that's a really good question. So that's one of the things, we haven't done that yet. But checking out the differences in cell polarity after these different perturbations is really important, because we know that these cells are very polarized in their epithelial phenotype, right? So in the neural tube, cells are polarized, and then they have to get rid of that polarization to actually round up and then collectively migrate out. And so one of the things that I'm sort of hoping my students will do is something that a lot of developmental biologists are starting to do, which is maximize the data. So we have all these beautiful images, and we can literally quantify differences in, protein localization and cell shape. And so that's what I'm hoping to do in the future, but we haven't done it yet. I do know that, again, with the TUB3 stuff, it's very clear there's cell changes and cell shape changes, but with the transcription factor stuff, we haven't identified that yet.
Yeah, that's exactly what came to my mind too. Is there's a wealth of data in your videos. They're amazing, and to get more out, that'd be amazing. That's really cool. Thank you.
Yes, I'd have a quick question just on the migration front, and maybe this just doesn't happen in the neural crest, but what role is there a role in the neural crest cells for paxillin? Because I believe that also sort of stabilizes adhesions, and adhesions you have to break those up in order to enable a cell to actually migrate. So is that something that you've taken a look at all?
I haven't yet, but I just got back from the Cell Contact and Adhesion Gordon Research Conference, and I was introduced by so many different cell-cell adhesion proteins that I had literally never considered and that haven't been studied in neural crest cells. So it's possible that paxillin has. If so, that would have been in frog, but it hasn't been studied in at least the amniotes that I've studied. But some of the frog people have done some true biochemistry and cell biology that we haven't done. But I'm really interested in expanding and thinking about, desmosomes and also like integrins and the interactions between the cells and the extracellular matrix. I think there's a lot to do there that we have not yet embarked on. And I think part of that was that we didn't have appropriate antibodies to look at these at the protein level, because, looking at an extracellular matrix gene or transcript is not the same as looking at fibronectin and collagen and all of that stuff. So we now have the tools to do this. And so I'm super excited about starting that. But no, we haven't looked at paxillin, but a paper just came out on Twitter talking about paxillin and a new role. And I was like, I have to read this.
Great, great. And you have such beautiful models too. So be interested to see.
Holly?
Oh, hi. Hi, Crystal. Beautiful talk.
Thank you.
I'm really taken by how you're able to image these really sensitive embryos in all these, in different species during this really critical time period. Over what time are you able to image, and how do you make that happen?
Yeah, so I would say that with the avian species, so our birds, they are the best imaging model for us, because in zebrafish cells, for example, beautiful imaging model, but neural crest cell development is so rapid in the cranial region anyways, that it's harder to catch. But with our models, with the birds, we have hours to, so we can do explant assays or do whole mount live imaging, and it's about I would say 3 to 5 hours to watch cells leave the neural tube and sort of migrate around. And so it's reasonable in terms of how big the data is and how big the videos are. but it's fast enough that we can do it, but it's slow enough that we can capture the different cell dynamics. But in our fixed samples, the sky is the limit. The only limitation we have with our fixed samples is that different fixation methods are necessary for different antibodies. But with the birds, so the proteins are so conserved that we generally use commercially available human and mouse antibodies, and they 90% of the time they work. And so we can really visualize some of these things that other models have challenges with. And also because they develop outside of the mother, you know, and we can just dissect out the embryos, so we don't have to worry about harvesting embryos from like a mammalian model.
Yeah, and then we had a question in the chat early on, Andy Lau, and asked, could you please also introduce your major methodologies and technologies in the lab? I think you had gone over that pretty extensively, but is there anything you want to add, any specifics to that?
So I think if this was at 914, Andy probably missed, we use a lot of fluorescence microscopy. So we use like Zeiss apotome sort of for our sections to do optical sectioning. We've been starting to use light sheet to get more 3D visualizations of our neural crest cells. Our students are using confocal imaging to do some of the higher mag, higher resolution stuff. But in terms of functional assays, we do a lot of stuff we can visualize. So we use in situ hybridization chain reaction, or HCR, to do multiplexed analysis of transcript localization and expression. And then we use immunohistochemistry with antibodies to do our protein localization stuff. And then we can do functional perturbations by using injection and electroporation in avian embryos ex ovo, so outside of the egg. We can inject and electroporate DNA and morpholinos. And so we can do these functional experiments really easily. I hope that answers your question.
I guess I have a quick question too about the magnitude of the data that you have to handle, and how, you know, how do you handle that? Do you move things around on thumb drives, or is it all connected to a nice sort of centralized data storage?
So, data storage is really tough. In theory, we would have, you know, yes, so that is an issue. We do have some main computers where data is stored, but it's true that everybody's roaming around with external hard drives because, you know, with the images, we get hundreds of images per data set, and so they're heavy too, so we can't just use flash drives for anything but like independent projects. And so everybody's roaming around with their own external hard drives that are like a terabyte and hoping that they don't get damaged as they stay on there. And honestly, using the light sheet and the confocal stuff is even heavier. So I think there's, you know, we have big issues in imaging with data storage and accessibility. And I'm curious to see how it's going to work with the new NIH mandates for, you know, data sharing in terms of imaging. This is going to be much harder than, you know, sharing sequences from sequencing. It's going to be a challenge to get all of that where it's supposed to be. So we struggle with it. I have my own, I have like a backpack full of hard drives where I'm just like taking everybody's data. So I always have a backup of it. But they each have their own as well. Yeah. I don't want to connect anything to a network because I'm afraid of viruses. I'm a PC person, and all of our microscopes are on PCs. And so, you know.
Yeah. It is a big problem to be grappling with. And I think that it is something that's coming to the forefront, particularly now that as you say, the NIH requirements have come out and everybody's being expected to make it all available. So yeah, just being a bit of a devil's advocate there, just inquiring.
Absolutely.
Any other questions in the audience?
Good to know. Feather says there's a working group for data management. So
yes,
there are so many cool working groups. By the way, all of the stuff that Vanessa presented in the beginning, I had no idea you guys were offering all of that. So I need to definitely share with my trainees. A lot of those things are phenomenal.
We have a great professional development program, and we have an exchange of experience program. So yes, if anybody in the audience is not already a BINA member, please join. We've got some fun stuff coming out in the next month. We have our annual image competition coming up, so that will go in the August 1 newsletter, and I think the deadline will be early October. But there's always an award for the top 3 photographs, and then the facility that produces the most submissions. So those are cash prizes. Please consider taking a look at the website because that'll be up on the website. But as I say, if you, or Vanessa mentioned at the beginning, if you aren't a member, if you join, you just get the one newsletter a month. We don't bombard you with information, but it's packed full of useful stuff. So.
Well, I can see there are at least a few people from my lab here, and then Ingrid, who also runs our imaging facility. So I'm sure that we're going to be participating.
That would be great. Yeah, please do. The more the merrier.
Well, thank you. This was really fun.
Great.
Yes,
It was a
thank
lovely
you so
presentation.
much.